So this entire unit is about cells, really. But before we sort of can get into cells, we need to focus on the microscope a little bit. So we start off this unit with a little discussion of different types of microscopes, how we use the microscope. Then we'll do some practice, do some labs. Then we'll move into cells, talk about cells, look at cells under the microscope, and so forth.
This is 7th or 8th?
This is a 7th grade unit. And the microscope section of this unit will be on your benchmark. So we'll see all these different things as we go through this unit. So there's some vocab here for your reference later on. When we use the microscopes, we're going to use two of the types of microscopes. And the two types of microscopes we will use are this type and the type back here. So this one, the one we'll use more frequently. This is called a compound light microscope. It's called compound. What is a compound word? Like two words combined. This is called a compound microscope because there's two different lenses that magnify the image as you're viewing it. Our microscopes can magnify from 40 times to 400 times, these compound microscopes. Others can go higher. Some can go up to 1,000 times with a special oil immersion lens, but our microscopes go up to 400 times. The other type of microscope we'll use is this type. This is called a dissecting microscope. It gives a lower level of magnification. Ours can magnify between 10 and 40 times.
What table would we use when we look at the cells?
We'll use the compound microscope. And then there's a type of microscope that we actually don't have here, the electron microscope. So there's a variety of what we call electron microscopes, scanning electron microscopes, tunneling electron microscopes. But these microscopes can magnify up to 1,000,000 times. You can actually see individual atoms in some types of electron microscopes. We don't have an electron microscope here. They actually do have one at Whitesboro High School. I did a workshop there last year. I took this image using the electron microscope. This is of an owl feather. Like these are the individual little fibers that make up the feather of an owl. It's really, really neat to see. But they have a special room dedicated to it. It looks more like a computer than it does a microscope. It's not something you put on a desk. It's a whole big unit that's, as big as this front desk maybe.
My friend said that they were doing a microscope here before, and that what is the one that you can see the cells like moving inside of it?
A compound microscope, you can see cells moving. So, yeah, detail?
Oh, that one has to be plugged in.
It's just different. These are newer. We just got these this year, and they have a battery-powered light source. These don't. They just have to be plugged in. So just to give you an idea about what things look like. In the compound microscope, so when you turn this on, light shines from the bottom through the stage and then through the object and into the eyepiece. So do you think, can I look at my hand through here?
Yeah.
Why not?
It's too big.
It's too big. Light's not going to pass through. So for things to be viewed through the compound light microscope, they have to be pretty thin enough to allow light through. And so generally the things you see in a microscope are very, very thin, and so you get a three-dimensional sort of view of the object. We will look at all three of these organisms later on in the year. They're all protists, single-celled freshwater organisms. But those are some examples of images from the compound microscope. When you look at the dissecting microscope, what's the first difference you notice about it?
It has two eyes.
Yeah, two eyepieces. Two eyepieces allow that, along with the light, allow you to see thicker objects and allow you to see three-dimensionally. You can see the depth of the object, which makes the view quite different. The dissecting microscope has a light that shines from above, So could I look at my hand under this?
Yeah.
Yes, the light bounces off my hand through the lenses, and because I have two eyepieces, I get good depth perception. You know, you need two forward-facing eyes to get good three-dimensional vision. So you could see things, and this is what something might look like under the dissecting microscope. You can look at things that are alive. They don't have to be sliced very thin. They don't have to be transparent. So it just allows you to see sort of different types of objects. Yeah, now the electron microscope gives you a very high resolution and great magnification, but the objects need to be treated in a certain way and coated in metal, and you need this complex equipment to see them, but they give you a really neat view of objects. Okay? And so here are some neat ones. This is a spider under the electron microscope. And it's pollen grains, human hair. These are all real electron microscope images. Now it gives a black and white view. Sometimes they're artificially colored by people after the fact to make things stand out or look better. Like these are artificially colored. That's a flea, cat's fur. That's a needle and thread. A maggot. Bunch of insects and stuff.
Is this actually real?
These are all real, yeah, these are all real.
But the colors on them,
The colors are not real, but
like on them, or the outside?
either. Spider, ants, grasshopper, fly. All right. So here we have our compound microscope. One of the things you're going to have to know are the parts of the microscope. We'll start this today. So the part you look through, it's called the eyepiece. It magnifies the image. There's a lens in here that helps make the image larger. The other lens that magnifies the image is down here. These are called the objective lenses. And our microscopes have three different ones. We call them low, medium, and high power. We can rotate this round piece to select from one to the other. This round thing we rotate is called the nose piece. Important thing to remember is your lens has to be clicked in. Like when I rotate this, like if I looked at it now, I can't see anything because the lens isn't lined up. It'll click into place once it's in the right position. The body tube is this section here, this long sort of tube. On this microscope is straight up. On our microscopes it's at an angle. But this helps keep the object the proper distance away from the lens to form a well-focused image. On the bottom is the light source. Ours are LED, and they're cordless. They have a battery which powers them. This microscope, it shows like an old-style microscope. What is this?
It's a mirror.
That's a mirror. And they used to have a separate light you plug in. You aimed it at the mirror. It reflected light up through the stage. We don't really use those anymore.
But couldn't you also make the light more powerful by doing that?
You could adjust it, but some microscopes have adjustable powers for the light. Where we put the object, this black flat part, is called the stage. And these little clips that hold the slide down, because you always are looking at slides when you're doing this, something mounted on a piece of glass. The thing that holds the slides down is called the stage clips. Talia?
Why are there three different ones?
They're different powers, low, medium, and high power. Different levels of magnification. Dylan?
Will we have to remember all of this?
Yes. Underneath the stage is a little lever called the diaphragm, and that helps regulate how much light is coming through.
And is it the size of how much like the
Yeah, it's like a little opening. It gets smaller or larger when you twist that. There's the arm, is this part like you would hold on to. And then on the bottom is the base, the sort of heavy thing it rests on. And then we have two really important knobs on this. You see them here. There's this large one and then the smaller one. They both basically do the same thing. They focus the image. This is called the coarse adjustment. And if you see how it moves the stage, it moves it a lot. You can actually see it moving up and down. The smaller one is called the fine adjustment. It moves it much less. All right, we'll use some microscopes tomorrow. So I'm glad we got through that.